Hello and welcome to the April 10th episode of APHA's 15 on COVID-19 series. I'm Dan Zlott, Vice President of Education here at APHA, and today we're going to be talking about COVID-19 testing. And boy, is this a hot topic. You may have heard the news, but the U.S. Department of Health and Human Services recently authorized pharmacists to order and administer COVID-19 tests. So this is huge news for the profession of pharmacy. Very exciting. So without further ado, let's go ahead and jump right in. I have nothing to disclose. Here are your learning objectives for today. All right, so why is COVID-19 so important? These are going to be pretty obvious. First and foremost, to identify the people who have the virus. Obviously, we want to prevent spread of the virus by those individuals. And later on down the road, once we're past this acute surge, we think that testing is going to help us play an important role in contact tracing once this surge phase is over. It's also important to identify people who've already been exposed to the virus and maybe are on the other side of their viral exposure. And this may be a really important factor in our efforts to return to something resembling normal life. And especially for healthcare workers, where it would be really comforting to know that you've already been exposed, you've developed antibodies, or maybe you developed some form of immunity, and so it's relatively safe for you to be exposed to COVID-19 patients, a huge factor for healthcare workers as well. So we'll be talking about all of this and how these tests work in today's episode. All right, so how does COVID-19 testing work? Well, generally speaking, the COVID-19 tests fall into one of three categories. The first one is testing directly for the genetic material of the virus. This is often referred to as molecular testing. I'm not a huge fan of that term. As you'll see, we're going to be using a lot of molecules in some of the other testing forms, and so it doesn't make sense to me, but the world uses that, so if you hear the term molecular testing, they're generally referring to looking for viral genetic material. You'll also hear this referred to as PCR, or polymerase chain reaction based testing. Another way of looking for COVID-19 is to test for antibodies against COVID-19. And so this is sometimes referred to as serologic testing. The antibodies tend to live in our plasma or sera, and so hence the serologic testing label. And then lastly, enzyme-linked immunosorbent assays. And these are kind of interesting. You may have been hearing a lot about these in the news. The coronavirus task force was asking hospitals and universities to develop these ELISA-based tests to accelerate the ability to test individuals. And so we'll touch on that as well. The nice thing about these, as I said, they're incredibly flexible, and you can test for just about anything as long as you can find the correct target, if you will, and then find something to label it with. And again, we'll go into detail on how this works in just a little bit. So let's start off with testing for viral genetic material. As I mentioned previously, this is usually done via a polymerase chain reaction based test. And so what we do is we take a piece of genetic material and we amplify it, or we copy it over and over and over again. And every time you run a sequence of the amplification, the number of copies of the DNA double, if you will. So you may be thinking, well, I thought coronavirus was an RNA virus. Why are we talking about DNA? Well, it turns out there's a simple solution to that. There's something called reverse transcriptase that takes RNA and converts it into DNA. So that's the first step. Once you have the complementary DNA from the RNA, you take that complementary DNA and you put it into a PCR, and it just copies it over and over again. So as I said, each round of amplification doubles the copies of genetic material. So you start off with 1. On your first round, you get 2, and then 4, and then 8, and then 16. You guys get the idea. It's a very quick way to get a lot of genetic material so that then we can try to do some interesting things with it. So what are those interesting things? Well, once it's been amplified, the next thing is to expose it to a probe, and that's our way of looking for specific sequences. Because of the nature of DNA or RNA, we can figure out the exact sequence of the genome of the virus, and we've done that, and so we know exactly what it looks like. So it's easy to create a matching probe that tests for the presence of that DNA or RNA. For the case of PCRs, it's DNA. And because of the uniqueness, we can say with high certainty that if our probe sequence is present, if we see it, we know that the viral genome is present. So the way we do this is we make probes that are labeled with a fluorescent marker. And it's kind of interesting. If you look at the photo on the slide, you see that before they bind to DNA, they sort of have like a hairpin-like shape, and they've got two markers at the end of them. One is a fluorescent dye, and the other is a quencher, and all it does is it basically stops the fluorescent dye from fluorescing when exposed to UV light. Well, when that probe binds to the target DNA strand, it flattens out, and the quencher molecule is separated from the fluorescent probe, and then when you shine UV light at it, it fluoresces. So after all this amplification and exposure to the probe, when you shine UV light at your sample, if you see fluorescence, you can be positive that you've got viral RNA, or viral DNA, in this case, present in your sample. So that's how these machines work. It's pretty cool. At least if you're a nerd, it's pretty cool. All right, so as I mentioned, the test is looking for that fluorescent signal. If one's present, you can be reasonably confident that your patient has COVID-19. There are some limitations to genetic tests. The tests only confirm the presence of viral genetic material. They don't confirm the presence of viral particles. So there are rare instances where people have had genetic material from the virus hanging around, but they can't confirm the presence of infectious particles. So relatively, probably a minor point at this point, but sometimes that can happen. We haven't heard about it so much with COVID-19, but there are instances in other diseases where that's come up. The other big thing is that a minimum copy number is needed at the beginning of this whole process in order to get accurate results. So when it comes to specimen collection, if there's not an adequate sample, if you don't get enough viral copies, when you run through this whole amplification process, there's not enough DNA at the end for the machine to detect the fluorescent signal. And so that can give false negative results in case you don't get an adequate sample. So that's another potential limitation. These tests can be complex to run. The machines that they've been coming out with in the last couple of weeks are really emphasizing ease of use and to the point where they're can be available as a point of care test in a pharmacy potentially. And so that takes a lot of the complexity out of it. However, the traditional way to run this test is a more complex process. What types of specimens can you use? Nasopharyngeal swabs, oropharyngeal swabs, nasal swabs, nasal washes, sputum, and bronchoalveolar lavages, or BALs if you happen to be in a hospital. Those are all reasonable samples. Nasopharyngeal swabs are probably the gold standard for now, but that may change over time. All right, let's talk about antibody tests for COVID-19. There's been a huge amount of interest in these as well. And these are all based on the idea that antibodies are specific, and so they bind to certain antigens and only those antigens, and we're going to try to take advantage of that. What we're going to do is basically tag a COVID-19 antigen with a marker of some kind, expose blood, serum, or some other fluid which contains antibodies to the tagged COVID-19 antigen, and then basically find a way to capture the antibodies and see if that tag is present. So let's talk about how this works. And this is kind of what you've been maybe seeing a lot, these types of little cassette-based tests that look like almost like a glucometer-based test. So let's actually see how these things work. So when inside of that little cassette, you have a little strip, and that strip is made up of 4 separate areas. There's a sample pad, a conjugate pad, a nitrocellulose reaction strip, and an absorption pad. That's just sort of the sponge to pull everything in. So let's say we have a drop of blood that we want to test. Inside that blood, of course, we have different kinds of antibodies, right? So we've got maybe a COVID-19 specific IgM antibody, a COVID-19 specific IgG antibody, other IgM antibodies, so not COVID-19, and then other IgG antibodies, again, not COVID-19. And so what we'll do is we'll go ahead and take that sample and we'll put it on the sample pad. The sample will begin to flow towards the absorption pad, and along the way it's going to encounter a couple things. On the conjugate pad, it comes pre-loaded with a COVID-19 antigen, as well as, and that COVID-19 antigen is bound to a tag or a marker molecule. Along with that, there's a non-human antibody, typically an animal antibody, rabbit most commonly, but sometimes mouse or other animals. And that antibody is also bound to the same tag or marker molecule. And this is going to serve as our control, as we'll see. So as a sample continues moving along, it hits the nitrocellulose reaction strip, and there's 3 pre-labeled spots on this nitrocellulose reaction strip. The first spot contains anti-human antibodies, and those anti-human antibodies are able to recognize human IgM. Doesn't matter what kind of IgM, they just grab all the IgM that's in that sample. So the second one is anti-human IgG. So same idea, but instead of grabbing IgM, it grabs IgG. And then lastly is this non-human antibody, this non-human antibody. So whether that's anti-mouse, anti-rabbit, etc., it's going to grab our control molecule and serve as the positive control. So as the fluid flows along, any IgM antibodies are going to stick on that first test spot there. And if there's no COVID-19 antibodies present, you'll just see IgM there, but there's no marker associated with it, so nothing happens. On the other hand, if there are COVID-19 IgM antibodies, they are going to pick up that COVID-19 antigen that's labeled from the conjugate pad on their way past, and they will bind to this spot, and they're going to bring that marker along with them. And so all of a sudden, we see marker showing up at that spot, and a little pink stripe appears. Now, most commonly, interestingly enough, the molecule that they use is actually gold. So that pink color actually comes from gold molecules. So the same thing happens for the IgG portion. The IgG gets trapped. If there's no COVID-19 present, no COVID-19 IgG present, you don't get a signal. If there is, it grabs that marker molecule, brings it with it, and you get a stripe there. And then, again, the control just rolls along, and it will bind to that non-human antibody, whatever that animal happens to be. And again, because it's tagged with a marker molecule as well, you get a control stripe showing that the fluid made it all the way through, the antibodies transited along the strip in the way that they should, and so that's your control line. And so that's how we determine whether you've got IgG, IgM, or none of the above against COVID-19. So what are some test limitations here? Well, first and foremost, it takes time to develop an antibody response once you've been exposed to an antigen. The image here is actually an immunoglobulin response curve upon exposure to dengue fever, which is another viral disease. But as you can see, IgM is the first type of antibody that's produced upon exposure to an antigen. And it takes about 4 to 7 days to really start to ramp up, and it peaks about 7 to 10 days after initial exposure. IgG takes a little bit longer, and it's the antibody that provides long-term protection for us. And so it starts to ramp up 7 to 10 days after initial exposure, and it peaks 10 to 14 days after initial exposure. So what that means is if you get caught early on before you have a chance to develop the antibodies, you might end up with a false negative. You could be infected, but you just haven't had enough time to develop an antibody response. So that's one limitation to this. The other thing is it doesn't test for the presence of the virus. It's just testing for the presence of antibodies. So there's a possibility you could be on the other side of your viral infection, still have both IgM and IgG around. Again, keep in mind that IgM peaks at 10 days, but it lasts for even longer. You can see 14 days and maybe even as far out as 21 days, there's significant amounts of IgM present. Does that mean you're infectious the whole time? We don't really know, but you might have a false positive where you've cleared your viremia, but now all we're seeing is the antibody response that remains. So one potential limitation there. Typical specimens, here it's going to be blood. Potentially down the road there may be other bodily fluids, but for now, blood. All right, let's talk a little bit about ELISA-based testing. So ELISA tests are limited only by really what you can target and what you can tag. They're incredibly flexible, and one benefit to them is that you can quantify your results. So the PCR-based testing we talked about, there's also the ability to quantify results there. That tends to be more in lab-grade machines, not so much in the point-of-care-based testing that you may have access to in a pharmacy. That's really going to be more qualitative. Yes or no. Same is true of those fingerstick antibody tests. Those are primarily going to be qualitative, again, giving you yes, there's IgM or IgG present, or no, there's not. It's not going to tell you how much. ELISA tests offer the ability to quantify how much is there. And because of the incredible flexibility that they provide, you can test for lots of different things. So the photo here is actually an ELISA plate. That's an assay plate. And note the wells in there and the different colors. We're going to go into that and kind of show you how that works. So here's one of those wells on an assay plate. So we've kind of zoomed in and we're looking at it from the side. Now on the bottom of the well, there's stuff that's stuck to it. In this case, we'll talk about COVID-19 antigen. So let's say we wanted to test for the presence of antibodies using ELISA. How do we do that? So start off with the well, and here's our drop of blood that we want to test. So within the blood, we've got plasma proteins, COVID-19 IgM possibly, other IgM antibodies, other IgG antibodies, so a whole bunch of stuff. So let's go ahead and throw that into the well. So we toss that in there. Any COVID-19 antibodies are going to stick to the COVID-19 antigen that's stuck to the bottom of the plate. So now we've got, essentially we've glued the IgM antibodies, the COVID-19 IgM antibodies to the bottom of the plate. We would go ahead and wash to get rid of anything that doesn't stick. And then now what we're going to do is introduce another antibody. So this is an anti-human IgM antibody. So it's designed to target human IgM. Doesn't matter what kind or what the target of that IgM is, it binds to IgM antibodies. And linked to that is an enzyme. And that enzyme is going to play an important role in just a second. So we would wash this off again. And in this case, where there is antibody present, it would stick. And then we're going to add an indicator molecule. So this is a molecule that's a substrate for the enzyme. And so if the enzyme is present, it will undergo a change and change colors. If the enzyme is not present, nothing will happen and the plate will just stay clear. So in the case where the enzyme is present and there's antibodies stuck to everything, when you add that indicator molecule and let it sit for a little bit, you get a color change in the well. And remember some of the blue wells versus the orange ones? That's exactly how this happens. So here we could say that there are definitely IgM antibodies against COVID-19 present in this sample based on the color change. So let's take a look at another example. As I said, ELISA tests are very flexible. So in this case, let's say we wanted to look at whether there were COVID-19 virus particles present. So we've talked about some of the limitations where maybe we can't tell whether or not there's actually virus present in a sample. So maybe we had an ELISA for that. So again, we've got a well on the assay plate, and this time the plate is coated with an animal COVID-19 antibody. So it comes from, again, mice, rabbits, something like that, and binds to COVID-19. So again, we've got our blood sample. In the blood, we've got the same thing, plasma proteins, maybe some COVID-19 IgM antibodies, other IgM antibodies, IgG antibodies, etc. So the only change is now we've got some virus particles present in the blood. So let's go ahead and now dump this in the well and we'll see what happens. So again, everything's going to go in there and mix around. The virus particles, if they're present, are going to bind to that animal antibody that's stuck on the plate. We'll do a washing, remove everything that doesn't stick, and so now we're left with just virus particles stuck to those antibodies. So just like before, we'll put in our indicator antibody. So this is an anti-animal antibody. In this case, it's linked to an enzyme. And here what we would see is because the binding site on the animal antibody is occupied by the virus, maybe the indicator antibody doesn't stick. When we do our washing, it goes away. So then we put in our indicator, again, the enzyme substrate, but there's no enzyme present to create a color change. So nothing happens. And all you're left with is your original color. Now, let's go back and run the other scenario. Let's say there's no virus present in this case. So we do the same thing. We run everything through, but this time when we put our indicator antibody in there, it's able to bind to the animal antibody that's stuck on the plate because there's no virus there occupying the receptor site. So at the same time, now when we go ahead and add that indicator molecule, there are enzymes present, and those enzymes change the molecule, change the color of the the solution in the plate, in the well, and you see the color change. So in this case, a color change would indicate no virus present, and a lack of color change would indicate the presence of virus. So you can see there's a lot of flexibility here. You can test for all kinds of different things, and you just have to be creative in terms of coming up with these ELISA tests. And so a lot of hospitals will be doing this and using these tests to confirm the presence of antibodies for their healthcare workers, et cetera. So what are some of the limitations of the test? Well, it's a high complexity test. It's not something that's easy to run, probably not something you'll be doing in your pharmacy. But if you work in a hospital, there's a decent chance that you may be subjected to an ELISA-based test to see if you've been exposed or if you have antibodies, et cetera, against COVID-19. It typically requires laboratory expertise, equipment, trained personnel, and significant validation of the tests before you start to use them clinically. So again, outside the scope of what you would normally see in a pharmacy, but we thought it was important to cover again, given the fact that lots of you do work in hospitals and may be subjected to ELISA-based assays to see if you have protection or not. Typical specimens here, pretty much anything, any bodily fluid or tissue digest that can be suspended in a liquid media is fair game for ELISA tests. So incredible flexibility. So we know there's a lot of questions out there about testing, about COVID-19 testing, about the new HHS guidance. APHA has a testing resources guide that we think is very, very helpful, and we hope it's helpful to you. It covers things like which tests are approved under emergency use authorizations, how to identify fraudulent claims regarding tests, and they are rampant right now. You guys are probably getting calls. If not, don't be surprised if you do. Links to the CDC and FDA testing resources, information about how CLIA applies to tests approved under EUAs, all of things we didn't have time to cover in today's 15 on COVID-19 episode, but do check out that link if you need more information about COVID-19 testing. Great resources there. So that will wrap up today's episode of 15 on COVID-19. We hope that was helpful for you in terms of understanding how these tests work and some of the limitations of the various tests. As always, our goal is to provide you with the education that you need to answer the questions that you're experiencing. We have been consistently getting some phenomenal questions, so thank you so much to those of you who have already submitted suggestions. We'd like you to continue doing that. covided@aphanet.org is the email address. Our topic for Monday is going to be convalescent plasma in COVID-19, and that's a question that we got through the suggestion box. So please keep them coming. Hope you guys have a great weekend. Please stay safe, and we will see you on Monday.